Okay, thank you everyone for attending this session. In this talk, we will discuss the strategies and the solutions for the discovery ADC candidates. Uh, During this presentation, I will first briefly go over the concepts and the technology trendings of antibody drug conjugates. Then we will have a detailed discussion on two crucial aspects of ADC discovery: how to find the suitable naked antibody leads, and how to evaluate the ADC candidates from the perspective of pharmacology effectiveness. Antibody drug conjugate is one of the fast growing modalities in recent years. It consists of three parts. The antibody is a highly selective carrier for tumor-associated antigens that has differentiated expression level on cancer cells versus normal tissues, while the linker works as a switch to release the killing agent in designated environments. The payload is a chemical compound with high systemic toxicity designed to induce target cell death after being internalized into the tumor cell and released. When you put these three pieces together, it can guide the toxin to tumor tissues and kill the cancer cells. Comparing to traditional chemotherapies, ADC demonstrated unique features to avoid systemic exposure to the toxins, leading to less adverse effects. Currently, there are 15 ADC products that have been approved globally. It is half and half for solid tumor and blood tumor. In addition, there are more than 200 pipelines in clinical trials and another 200 plus pipelines in preclinical stage. United States and China are the biggest cancer therapeutic markets and takes about 80% of the global pipelines. So far, there has been three generations of ADCs. The first generation is comprised of traditional chemotherapy drugs and non-cleavable linkers. The major issues of these ADCs include undesirable efficacy from the toxin, unstable linker leading to systemic toxicity and a narrow dosage window, hydrophobic toxin leading to negative impacts on the uptake of the drug. To overcome these weaknesses, people improved the toxin payloads in the second generation of ADC, but still relies on random conjugation, which makes it challenging to achieve homogeneity in terms of the DAR, number of payload molecules on each molecule of antibody. Nowadays, in the third generation of ADC, site-directed conjugation is widely used to overcome the DAR homogeneity challenge. And the concept of ADC has expanded from traditional chemical toxin to various uh, modules such as peptide, oligonucleotide, radioisotope, and even protein. In order for the ADC to kill the cancer cell, it needs to be transported into the target cell in which the toxin will be released. This process is called endocytosis or internalization. This unique feature brings several challenges in ADC discovery, including tissue penetration, target specificity, internalization efficiencies, timing of a payload release, the killing efficacy of a payload, etc., etc. As a result, a panel of factors needs to be considered during the discovery stage, such as choosing a suitable target with differentiated expression level on the cancer cell versus normal tissue, high affinity antibodies with desirable internalization capability, suitable linker and payload combinations, and also early stage pharmacology assessments for efficacy and safety in cells and in animal models. Next, we will move on to the first part of ADC discovery strategies, how to identify desirable naked antibody leads for ADC applications. So this is a general workflow of ADC discovery. Once you decide on the targets, a variety of technologies such as hybridomas, single B cells, phage libraries can be used to generate antibody leads. In the meantime, selection of a linker and a payload will also be made based on the indications, disease biology, and the MOA. Considering the potential safety risk, lots of people tend to choose toxins with plenty of clinical track record instead of those really novel ones. After obtaining antibody leads with desirable internalization features, a panel of ADCs uh, will be prepared in small scale, followed by cell-based assays and animal studies. At this point, the candidates will be narrowed down to less than half a dozen. These top candidates will go through antibody engineering, including humanization, affinity tuning, and FC engineering to gain particular features with optimized profile. Finally, in vitro and in vivo pharmacology model will be utilized again to validate the efficacy and the safety of the engineered final sequence, followed by developability studies to assess the product stabilities and homogeneity. The functional evaluation of ADC consists of two major aspects. One is the antibody internalization assay from the antibody FAB region and the cytotoxicity from the payload toxin side. During the naked antibody discovery stage, we will first focus on the internalization parts mediated by antigen binding to the FAB region. The first option is live cell imaging based internalizations. So basically, it utilizes a hardware called IncuSite to visualize the whole endocytosis process through fluorescent signals. The key features of this equipment is to generate internalization kinetics with a time course. As you can see from the case study of anti-RR1 ADC in the bottom panel, for each defined concentration, for each defined antibody concentration of the benchmark UC961, there will be a curve reflecting the intensity of the internalization on the time axis. And you can include multiple curves to reflect the multiple curves in one graph to visualize the kinetics and the dose-dependent response. In addition, you can also generate a dose-dependent curves with fixed incubation time to calculate the internalization efficacy, the EC50s. Moreover, this assay can actually run with antibody supernatant, providing a high throughput approach to evaluate internalization capabilities during the early antibody screening stage. So by doing this, you can actually avoid losing potential candidates by making selections not solely based on the binding affinities. The second option is toxin conjugated antibody-based cytotoxicity assays. The DT3C is a synthetic toxin acting as a surrogate of a payload toxin in ADCs. Unlike typical toxin molecules, DT3C can associate with the Fc region of antibody through non-covalent interactions. After association with antibody, the whole complex will be uptaken by target cell, leading to the release DT3C toxin, which results in the cell killing. Since the binding between DT3C and the antibody is non-covalent, the chemical conjugation process, which is required for payload toxin in ADC, is no longer needed here. So basically, this assay is using the DT3C mediated cytotoxicity as a measurement of the internalization. Similar to the first option, this approach can also run in a high throughput fashion using antibody supernatants, providing a way to differentiate antibody leads in early screening stage. To demonstrate the robustness of this assay, we tested the DT3C system with the presence of a Herceptin and a negative control IgG on the right panel. As you can see from the graph here, SKBR3 cells gives a perfect dose curve of inhibition after incubation with Herceptin plus DT3C for 48 hours, while the negative control IgG, as illustrated as the black one, basically flat. The third option is pH indicator. The detection mechanism is also based on the co-internalization of fluorescence signal along with the antibodies, kind of similar to the first option, the incusites. The pH rodel consists of an anti-Fc Fab fragment, this one, and a pH-sensitive dye which become highly fluorescent only in acidic environments, such as those of cell lysosomes and endosomes. The case study we did with the Herceptin showed decent dose response curve of fluorescent signals after incubation for 16 hours. The fourth option is cell surface fluorescence quenching. So it is a pretty straightforward flow cytometry-based assay to compare the relative amount of antibody entering the target cells. To put it in a simple way, the antibody will be incubated with target cells in two conditions in parallel, 4 degrees and 37 degrees. And the amount of the cell surface binding antibodies will be measured by typical fluorescent secondary antibodies. The signal difference reflects the relative amount of antibodies entering the target cells. The rationale of this approach is first assuming there is minimal internalization under 4 degree, and it also intentionally neglects other factors contributing to the fluorescence signal change, such as dissociation of antibodies from the target cell. As a result, this approach is not as accurate as those previous ones, but it requires the minimal hardware and reagent support, which makes it still a very popular option for early stage screening. So with all of these four options on hand, people will, will naturally wonder which one should I trust, right? So to answer these questions, we did a side-by-side comparison of these four assets using from three to ADCs. Two of them are already launched, while the third one is still in phase two. Judging by the results, all four approaches gave similar ranking pattern of these three antibodies, and also similar range of EC50 values, except for the last one, the cell surface fluorescence quenching. This data demonstrated the reliability and the consistency between different assays. With that being said, you may still wonder which one should I choose. So we'll put together all of them in one table. The live cell imaging approach gives the best data and high throughput capabilities, which makes it the best choice for both larger scale screening and candidates characterization. The DT3C toxin approach is also capable of high throughput and relatively low cost, making it an alternative option. The pH indicator is relatively expensive, while the fluorescence quenching method is not as good as the other in terms of the reproducibility. Next, let's move on to the second part of ADC discoveries, the pharmacology assessment of the drug antibody drug conjugates. So once you made the selection of the naked antibody leads, the next step is the payload and the link conjugations. Currently, there are a variety of payload and link used in different ADC pipelines. Here at Genusco Probios, we can offer a library of chemicals, including more than 100 toxins and 700 plus linkers, as a reagent for proof of concept validation during the ADC discovery. In addition, you can also choose from traditional cysteine lysine-based conjugation chemistry or site-specific approach, site-specific conjugation approach to precisely control the DAR and the homogeneity. After the conjugation, ADC candidates will be first tested in vitro for cell killing efficacies. This can be achieved through viability assays, apoptosis assays, or cell cycle assays. The case study done with HER2 showed on the right panel demonstrate that both ADC test articles, one of them is the reagent version of S8201, both of them gives dose-dependent inhibition on the SKBR3 cells as measured by the cell titer glow reagents. In addition to direct inhibition, bystander effect is another MOA for ADCs. Bystander cells is the surrounding cells of the tumors, which could be antigen positive or antigen negative. When the tumor cells is lysed upon ADC function, toxin will be released and kill the neighboring cells. This MOA can be reconstituted in vitro as illustrated in the right panel. In the positive and the negative control group, testing ADC will be incubated with either antigen positive or negative cells, in this case, the HER2. And in the testing group at the bottom, conditional media from the drug-treated HER2 cells is transferred to the HER2 minus cell for efficacy evaluation. After all of these in vitro functional screening and validations, it is absolutely necessary to verify the efficacy and the safety of the ADC candidates in animal models. Typically, it includes three packages. Uh, for the efficacy part, people usually start with cancer cell line screening based on the indication and target expression profile, followed by tumorigenesis and efficacy tests. PK assay needs to be carried out both in vivo and ex vivo to determine the ADC stability, especially the linker parts and the controlled payload release. Toxicity studies is also a crucial part, including myelotoxicity assessment, tissue cross-reactivities, and payload-derived pathology. Here at GenScript Bio, we have established 24 tumor types with tumorigenesis from more than 70 cancer cell lines. In addition, we also provide 200 plus cell bank and a IHC panel for pre-study screening of a cancer cell line. PK and toxicity are crucial factors for the determination of ADC therapeutic windows, which is the constant import during ADC drug development. In the discovery stage, we usually pay attention to three checkpoints: the stabilities, target delivery, and the systemic toxicities. Stability can be assessed by ex vivo plasma stability tests and in vivo PK studies. Myelotoxicity assay using CD34 positive stem cell ex vivo. So at the end, let's come back to this process of ADC discoveries. There are a few key checkpoints that needs to be included. First of all, a high throughput internalization screening during the antibody leads generation stage will greatly help to weed out those non-desirable candidates. Secondly, pilot efficacy studies in animal models during early discovery stage gives us the opportunity to test the different combinations of naked antibody leads and payload linker in the actual disease model. Later on, after finalizing the candidate sequence, comprehensive PK/PD and the tox study, and also the developability assessments will greatly reduce the risk of failure in subsequent preclinical studies. So I think that will be all. Thank you everyone for listening. Let's move on to the Q&A session.
Thanks for Dr. Chen's excellent presentation. If you have any questions, please type it on the Q&A panel or chat panel. As you enter the questions, please also follow our LinkedIn account, GenScript Profile, and Korean audience can also scan this QR code and follow our Kakao channel. There will be more online sharing resources, and we will show them on our LinkedIn and Kakao channel at the first time. Okay, we received some questions from the audience. Let's take a look at the first question. Hi, Dr. Chen, can you hear me?
Yes, I see the question from the Q&A box. It's, can you please touch upon the selection of a linker, its influence on bystander effect? Thanks. So generally speaking, in order for a ADC to have a bystander effect, the linker itself needs to be a cleavable versions, which means the linker can be cleaved by certain enzyme or chemicals before entering the target cells with the TA expressions. By doing that, the, uh, the ADC will bring the toxins into the tumor microenvironment, and before entering the target cells, the payload will be released and start to kill the neighboring cells with differentiated expression profile of the TA antigens. I'm not sure if that's what you're asking.
Okay, thanks for your answer. Let's move to the next question. The next question is, what antibody candidate is prone to be developed as ADC?
So, do you mean what's
Antibody candidates.
preferred?
Yeah.
What is the preferred profile for a antibody to become a candidate for ADC, right?
Yeah.
Usually, the first criteria is the internalizations. In order for a ADC to be effective, it needs to be internalized efficiently into the cancer cell through the binding to the TA. So during the screening stage of the naked antibodies, we will conduct the internalization evaluation and the screenings and select those antibody candidates with desirable internalization profile, which means it's not necessarily to be fast internalization or strong internalization. It all depends on the target product profile, your disease, the indications, and also the TA antigens. But at least the naked antibody should be able to be taken into the target cells, which is the tumor cells, in a limited time windows.
Okay, thank you for your sharing. And the next question is, what is your recommended approach for assessing antibody internalization?
Actually, we did share a slide. Well, actually, it's a couple of slides talking about the internalization. Currently, we offer 4 different approach, and as I mentioned in the presentations, our preferred approach is either the in-queue site with the high throughput capabilities, or the DT3C, the toxin conjugated approach. Both of these 2 approach can work with either antibody supernatant or purified antibodies, makes it feasible to conduct high throughput screening and full profile characterizations at the same time.
Okay, thank you. And here is a question come from the Q&A panel. Any examples when bystander effect is not desirable?
Well, yes. So sometimes actually it depends on the cancer itself, the indications. Sometimes the TA, the antigen expression level inside the tumor tissues is not consistent. There are tumor cells with high expression, and there are tumor cells with low expression. Under these circumstances, the bystander effect is usually it prefers, so that to maximize the killing within the tissue, within the tumor microenvironment, regardless the expression level of the TAs. On the contrary, for some certain cancer tissues, if the TA expression levels is pretty universally high or maintained at similar levels, bystander effect is not really required.
Thank you. I also received a private question. The question is, what is your view on Chaudhary and Datopatamab Diluxcan product profiles? I also sent it through our Teams group. Could you please check this question and answer it?
Okay, what is your view on Chaudhary and well, if I remember correctly, the Chaudhary is targeting to CHOP2. CHOP2 is a pretty nasty target, TA target for ADCs. There are safety issues and those limiting issues. I think that's the major problems with all the ADCs targeting the CHOP2 targets. Well, it's a pretty big questions. I'm not sure if my answers is satisfying. I mean, people could just write a book regarding these two different drugs.
Okay, thank you. And the next question is, do you have any site-specific compound conjugation technology for the ADC?
Yeah, actually, we have a collaboration with a IP owner who is specialized in the glycan-based site-specific conjugation technologies from China, and it's a well-established and validated platforms. The most advanced pipelines with that technologies is already in the, I believe it's already in the clinical stage, in the preclinical or clinical stage. So so the answer is yes, and we're open for any further discussions if you're interested in this particular glycan-based site-specific conjugation technology.
Okay, thank you. Another private question from my side. There are pros and cons to increasing DAR, so considering production cost as well, can you tell us what is the most efficient DAR for ADC?
Well, definitely there's no universal answers for that. It's all up to the indications and the toxin, the payload you choose. For instance, for the DS 8201, it choose a less toxic payload so that the DAR has to be higher than the other common ones like the MMAE, MMAF. So, like I said, it really depends on the safety window of the payload itself and the disease itself.
Okay, thank you. Another question is DAR value important or payload potency to have reasonable clinical efficacy for ADC?
Sorry, could you repeat that question again, or could you type it in the?
Yeah, I sent it in the group too. Yeah, is DAR value important or payload potency to have reasonable clinical efficacy for ADC?
So is DAR value important or payload potency to have reasonable clinical efficacy for ADC? Of course, the efficacy of the ADCs is ultimately determined by the payload potencies, and in the meantime, the safety window is another key factors determining the overall potency of the ADCs. If it's really toxic, you won't be able to give it with a high dose, and that will limit the ADC potencies. And like I said in the previous questions, the DAR value has to be carefully considered and evaluated in accordance with the potency of the payload and the desired indications.
Okay, thank you. Thank you for your answers. That's all the questions we have for now.